Hello, I'm Kristin Snyder, a DVM PhD candidate in Bruce Walczak's lab. I'm very excited to share my work with you today, which is enhancing natural killer cell antibody-dependent cell-mediated cytotoxicity for the treatment of ovarian cancer. Ovarian cancer is a leading cause of cancer-related deaths among women worldwide. An estimated 1 in 78 women will be diagnosed with ovarian cancer in their lifetime. As the fifth leading cause of cancer-related deaths in women, ovarian cancer accounts for at least 150,000 deaths a year. Due to a lack of early detection methods and symptoms that are nonspecific, such as bloating, belly and back pain, and weight loss, most women are diagnosed with advanced disease, which is often in stage 3 or stage 4. In cancer, there is no stage 5. Advanced ovarian cancer is very complicated to treat. Standard therapies consist of debulking surgery, which aims to remove as much tumor tissue as possible, and conventional chemotherapy and radiation. A challenge currently facing the treatment of ovarian cancer is that many patients develop resistance to these standard therapies. When a patient develops resistance, their average life expectancy is only about 9 months. Therefore, new and innovative treatment strategies for ovarian cancer are desperately needed. In the last 20 years, immunotherapy has revolutionized how we treat cancer. Immunotherapy is a type of cancer treatment that uses the immune system to fight cancer. There are many types of immunotherapies, but the two that I will focus on today are adoptive cell therapies and monoclonal antibody treatments. Many of you have likely already heard of adoptive cell therapies, such as CAR T-cell therapy, which uses specialized cells of the immune system to fight and kill cancer. Monoclonal antibodies are infused into patients and include drugs like Herceptin, which specifically target proteins expressed on the surface of the cancer cell. Cells in the immune system then bind to the antibody attached to the cancer cell to kill the tumor. The goal of my project is to combine cellular immunotherapy with monoclonal antibodies to treat ovarian cancer. Our lab studies natural killer cells, also commonly called NK cells. NK cells are a specialized type of white blood cell called a lymphocyte. They circulate in the blood and tissues and play a critical role in eliminating cancer cells. One way NK cells kill cancer cells is through a process called antibody-dependent cell-mediated cytotoxicity, or ADCC. This occurs through a receptor called CD16A. CD16A binds to an antibody on a tumor cell, and then the NK cell will release specialized granules that poke holes in the tumor cell and result in tumor cell death. CD16A has a few issues that may limit the effectiveness of NK cell immunotherapies. First, CD16A doesn't bind antibodies very strongly. You can think of the affinity of CD16A for antibodies like a weak handshake. Secondly, CD16A gets cut from the surface of the NK cell when the NK cell becomes activated. Since our goal with NK cell immunotherapies is to activate the NK cell, this poses a big problem. To combat these issues with CD16A, we developed CD6416A, which has CD64 on the outside of the NK cell and CD16A on the inside of the NK cell. CD64 has a very high affinity for antibodies, which is a lot like a firm handshake instead of shaking hands with a fish. CD64 also does not get cut from the cell surface like CD16A does. We found that NK cells expressing CD6416A kill ovarian cancer cells when combined with the therapeutic antibody Herceptin. In this video, the ovarian cancer cells are green, and the NK cells are the tiny black dots moving along the screen. Over time, you can see that the NK cells target and kill the green cancer cells, which is indicated by the cells balling up and losing their green signal. We all know someone who has been affected by cancer. Immunotherapy has changed the course of cancer treatment, but it is still in its infancy and can use further improvement. An advantage of our NK cell platform is that it can be combined with many different therapeutic antibodies to target cancers other than ovarian cancer. Additionally, our findings may be useful across species. What we learned from NK cell immunotherapies designed from humans may be translated back to veterinary medicine for treating cancer in our furry family members as well.